Hi, everyone. We are really excited to be back with the second round of our presentations for tonight. We have a whole new crop of students. They've all been working together all summer, but this has been a fantastic summer research experience. We have such a range of topics. You'll see a huge selection tonight, and these students really worked hard this whole summer. They started with an area of interest. They designed a whole project in order to answer their research question, and they went through all of the stages of figuring out what variables to collect, how to get that data, how to process it and turn it into something that they can analyze. They figured out the statistics to use, and they are very excited to report on their results tonight with a presentation, and we'll be finalizing manuscripts for publication over the next 2 weeks as we kind of finish up here. But we are super excited. Thank you all for joining us and coming to celebrate these students tonight. And we have a bunch of students. I want to start by having mentors introduce the different students because they are the ones that have worked with the students the most. And so first, I want to bring up Sean, and he is going to introduce our first presenter, who is Ben.
All right. Thank you, Megan. Yep, it is my honor to introduce Ben tonight. Just want to say a little bit. Ben was a joy to work with. His adaptability on this project, and it's just, it's a joy. He's a joy to work with, and he's going to be talking about microgravity and its potential uses.
Awesome. All right, take it away, Ben.
Hi, my name is Ben Katadi, and I'm going to be presenting about motion in microgravity. What is microgravity? Microgravity is a state in which the gravitational force is negligible on an object, and it can be achieved on Earth when an object reaches terminal velocity. Terminal velocity is reached when an object is in free fall and can be reached when an object is dropped from any height. The reason that gravitational force is negligible in terminal velocity is because the object cannot accelerate anymore because of the gravitational force, and thus the gravitational force is negligible. Hydrophobicity and hydrophilicity are two different forces that act on an object in microgravity. Forces. Hydrophilicity is the property of a substance to be attracted to water. Hydrophilic substances are usually polar, and this means that they usually have unequal charges. Then this is why they are attracted to water. Hydrophobicity is a property of a substance to repel water. Hydrophobic substances are usually nonpolar, which means they have equal charges, and they repel water due to their equal charges. Currently, studies have shown that hydrophobic substances repel water stronger, and hydrophilic substances attract water with greater strength in microgravity. One study in particular showed that in microgravity, water reached Kinsey states with hydrophobic substances. And this basically means that water repelled fully from a hydrophobic substance in microgravity. Another study showed that in microgravity, there was condensation from the water on hydrophilic substances, which shows that the water was attracted to the hydrophilic substance in microgravity. And that was just a short video, basically detailing how these forces are in action. And that was with tap water. So what did you just see? This graphic shows how the forces work. So the hydrophilic force pulls the propeller into the water, and the hydrophobic force pushes the propeller out of the water. And that basically creates the motion that you just saw in the video. Our research question for this experiment was, how can we create motion in microgravity through chemical means? And we hypothesized that if the hydrophobic and hydrophilic forces are used in unison to make deionized water, the motion created will be significant enough to be harvested to make energy, thus creating another way to make clean energy. These are the methods that we used for our experiment. Firstly, our propellers were coated with hydrophobic sprays and hydrophilic sprays. The hydrophilic spray we used in particular was polyurethane spray, specifically Minuax polyurethane spray. The hydrophobic spray we used was the Never Wet super hydrophobic coating spray. And the coating pattern we used was specific to maximize the forces, and it's seen in the bottom right hand corner. And we used a sort of checkerboard pattern to really maximize the forces and to make the forces work in unison. And the propellers were fixed into a plastic container, which was then filled with deionized water or tap water, depending on what test we were doing. And the test setup is seen in the graphic to the left. Secondly, we had to collect our data. The plastic container that with our propellers was put into a teardrop shaped apparatus to drop. And this was a teardrop shape to minimize drag. The height of these drops was about 14 feet. And you can see one of these drops, an example in the bottom right-hand corner, and an example of our apparatus that we dropped in the bottom left-hand corner. To record each drop, we also had a camera inside the apparatus to collect data. These drops got about 1.5 seconds in microgravity. And to analyze these drops and the data that we got from the camera, we used a linear regression model. These are the results of our microgravity tests. So these results were all averaged over 3 tests for each type of water. And as you can see, the variability in the graphs show the inconsistency between tests. And positive values in the graph indicate clockwise motion, while negative values indicate counterclockwise motion. And you can see that the tap water has way more variability than the deionized water, and it moves between clockwise and counterclockwise motion in some areas, which means that the deionized water is the more consistent choice. To accurately find the speed in which the propellers were moving, we use linear regression. With the tap water, the propellers spin at about 2.37 millimeters per second, and that's seen through the graph in the bottom left-hand corner. With deionized water, the propeller spun about 1.685 millimeters per second, and that is seen in the graph to the bottom right-hand corner. Our results were very correlated, as indicated with a very low p-value, less than 0.01. And this basically means that the time in microgravity and the motion created was very related. However, based on our test, the more practical choice is deionized water. And the reason for this is it's not inconsistent at all. And if we're planning to use this in real life, then consistency would be a high priority. However, since a limited number of tests were done and we only got a limited number of time in microgravity, more research is needed to make a conclusion. In society today, there's a very big need to make clean energy. And if this method is used, carbon emissions can be reduced significantly. So to earn acknowledgements, we want to acknowledge DVC, Diablo Valley College, a local community college here in San Ramon, for giving us deionized water for our tests. We want to thank another Headwaters mentor, Danny, for our edits. I also want to thank Megan for coordinating everything, and I want to thank my mentor, Sean, for all the hours he put into my slides and into my manuscripts.
Awesome, Ben. What a fantastic project. And Sean, I'll give you the first floor here. Do you have any questions for Ben?
Well, I do have a question. Yeah, it's something, Ben, you and I have chit-chatted about a little bit. But what do you foresee as far as, do you think that this kind of research can give us like multiple energetic uses as we push forward in this work?
Yeah, I think so. If the motion created is significant enough, then the plan is to send something into space where there is no gravity, and then we can make clean energy, kind of like a water mill.
Excellent, right?
Awesome.
I mean, how cool is that?
Very cool. I've got a question for you, and that is mostly were there any, and if there were, how did you address any struggles that you had in trying to figure out how to create a microgravity situation for your experiment?
Well, like firstly is like it was kind of just a lot of research on how to create microgravity and how to make prolonged periods of microgravity, and it was just developing like something that could have and be in microgravity for a long time. And yeah.
Awesome. Did anything surprise you that you found in either your background research or in your own experiment and your results?
Yeah, so for the tap water versus deionized water, for our results, strictly physics speaking, deionized water, since it has no ions and no other charges, there should be a larger force of attraction and force of repulsion for the hydrophilic and hydrophobic substances respectively. But for our results, as we saw, it was actually bigger for tap water than deionized water. So that was kind of surprising.
Cool. It's always fun to find something that you're not expecting. Sean, do you have any other questions for Ben?
No, not. I think he touched on everything. Like I said, it's something we sat around kind of at the beginning, right, Ben? We kind of talked about stuff, and it's like throwing out ideas, and okay, what are we gonna do? And well, what do you think DI water would do? And it was like, well, like Ben just touched on, right? Like it's DI water, so we should see a stronger push and pull, but results speak for themselves, right?
Awesome. Well, fantastic job, Ben. This is super interesting. And I don't know if anyone has ever done a microgravity project for a Headwaters research experience before. So that'll be something new, a new topic to add to our journal. So thank you. Great job.
Thank you.
Yeah, I'm going to keep Sean up here because next we have Anjali. And Sean, if you would introduce her and a little bit about working with her and her project.
Of course. So yeah, as we say, it's also my pleasure to introduce Anjali. Anjali has a great openness and eagerness and cleverness when it came to this project. The material and the work that she did, which I don't want to take too much from her stage, but studying water quality and phosphorus, which I'm going to let her explain.
Yeah, so thank you, Sean. So this was my project. It was on the effect of urbanization upon algae bloom formation. And in order to ensure the health of aquatic ecosystems and the organisms that interact within them, it is imperative to understand how human behaviors impact these habitats. So for a couple of definitions, just to get us going, urbanization is a population shift to urban areas or population growth in urban areas, and this goes hand in hand with industrialization or the formation of an economy into one that is more reliant on manufacturing. And eutrophication is the leading cause of freshwater pollution across the globe and is defined as the process by which nutrients accumulate in a body of water, resulting in an increased growth of microorganisms. And these nutrients enter bodies of water through a process called nutrient overloading, in which a certain quantity of nutrients enters an ecosystem over a period of time. And for what exactly algae blooms are, algae is an autotrophic organism, meaning that it produces its own food rather than consuming other organisms, and it's composed of bacteria. HABs, or harmful algae blooms, are the product of the rapid accumulation of phytoplankton, resulting in hypoxia and the perishing of aquatic organisms. Now, this equation just kind of shows you how these algae masses come to be. And as you can see, nitrate and phosphoric acid are two of the reactants in this equation. And of course, nitrate contains nitrogen and phosphoric acid contains phosphorus. And these are the nutrients we're referring to when we talk about eutrophication. As for my specific research question, I wanted to study the effect of urbanization upon phosphorus content and algae biomass, and I examined this through Weber Lake and Lake Natoma. And now before we get further into this, the reason I chose phosphorus content specifically is because it's understood that phosphorus is the limiting nutrient in freshwater systems, meaning that it is the nutrient that algae primarily relies on in order to form. And my hypothesis was that lakes with higher exposure to anthropological influence will exhibit higher phosphorus content in algae biomass, as these cities will expose these bodies of water to sewage runoff and general pollution. As for the two lakes that I looked at, this was Lake Natoma, and as you can see, it is heavily surrounded by urbanization. And in comparison, this is Weber Lake, which is much more secluded. And while both of these lakes were in Northern California, they both had vastly different exposure to anthropological influence, which is why I chose them. As for my methods, in order to prepare, I randomly mapped out 10 testing sites per lake, but as I was collecting this data manually, which entailed me on a paddleboard and all of my equipment, going out and paddling across these lakes and collecting data, in Lake Natoma, I was limited as this was difficult to get across the whole lake since it's a very long lake, but you can see that these are the places that I took samples at. And as for data collection itself, I collected water samples at 3 distinct depths as well as 1 surface sample, and then I acidified these with 0.1 molar HCl. And these samples were also refrigerated immediately after collection. And the reason I collected at 3 distinct depths was that in these systems, phosphorus and other nutrients are typically cycled through, so nutrient levels may be different at different depths. And this is a picture of me collecting my data, and this is the tool that I used. So I took a 12-foot long extendable window cleaning pole, and I attached a water bottle to the end of it with a cork and a string so that I could allow water to enter when I desired. And as for other data that I collected, turbidity is the measurement of the opacity or cloudiness of water. And this shows the level of particles such as sediment, plankton, or organic byproducts in a body of water. And this is important to my study as turbidity is loosely indicative of chlorophyll A levels, high amounts of which are characteristic of algae blooms. And what's pictured on the right is the tool that I used to collect these readings, and it's called a Secchi disk. And essentially what you do is you average the depth at which it disappears with the depth at which it reappears, and this gives you your Secchi value. As for water testing, I tested for phosphorus, which was measured in parts per million for every 50 milliliter sample. And I utilized water testing strips, and you can see in the image at the bottom right corner of the slide that this was on a blue scale, so shades and tints of blue, and then I could approximate how much phosphorus was in each sample. As for algae biomass, which was measured in grams per 50 milliliter vials, I strained the water through filter paper and weighed the dry mass of the algae. And before doing this, I removed anything that was like sticks or leaves or evidently not algae. As for my results, this figure compares phosphorus content to algae biomass, and as you can see by the orange regression line, there is a strong positive correlation between these two variables, and I got a p-value of less than 0.01 upon conducting a linear regression test, which again indicates strong confidence in statistical significance. And the implications of this are that as phosphorus content in these two lakes increased, as did algae biomass. Furthermore, I compared the phosphorus content at the two lakes, and as you can see, Lake Natoma has a much wider spread of data and instances of higher phosphorus content. But that being said, upon running a t-test on this figure, I got a p-value of 0.22, which indicates a lack of confidence in the statistical significance between these variables. And what this means for my study is that the difference in phosphorus content between these two lakes is not necessarily statistically significant. So if I were to do this experiment again, I might not get the exact same results. But that being said, upon adding depth as a variable into this figure, I came back with different results. And in order to do so, I separated my depths into shallow and deep and combined this with the lake at which the sample was collected and ran this against phosphorus content. And when I ran an ANOVA statistical test, I got a p-value of 0.03, which indicates strong confidence in statistical significance. I ran similar tests when comparing the lake's algae biomass values, and again, when it was just lake versus algae biomass, I got a p-value of 0.23, which indicates a lack of confidence in statistical significance. But upon adding depth as a third variable into this experiment, I got a p-value of 0.15, which indicates stronger confidence in statistical significance. The final test and comparison that I made was that of Secchi value. Now, it's important to note that Secchi value and turbidity are inversely proportional, meaning that, for example, in Lake Natoma, a higher Secchi value is indicative of low turbidity or better visibility, and this is contrary to what we might have hypothesized or assumed, as Lake Natoma had higher amounts of algae, and this figure had a p-value of less than 0.01, which indicated strong confidence in statistical significance. So now, as to discuss these results, first let's tackle the issue of turbidity. So the reasonings for these results are likely that at Weber Lake, I collected less deep water samples, which would influence the mean Secchi value. And additionally, Weber Lake is covered in, I think 60% of the lake is covered in submerged aquatic plants. And this results in high turbidity, so poor visibility, but also a cleaner system as these plants remove dissolved phosphorus from the water column. Additionally, Lake Natoma depicted much higher, phosphorus depicted much higher statistical significance in these tests. And the reasoning for this is likely that algae blooms take longer to form. And there's a lot of factors that go into their formation, whereas phosphorus is either there or it isn't. So when I added depth to these tests and phosphorus content still had higher confidence in statistical significance than algae blooms, that is likely why. As for things that I might change in the future, I would definitely like to explore Lake Natoma more, because as we saw, Lake Natoma had rather low amounts of algae and phosphorus content despite our hypothesis. But if we go to look back at Lake Natoma, you can see that there's a lot of green space around the lake, and we call this a green space buffer. Essentially what it does is it soaks up the nutrient content and the water that is coming in from the city, so sewage, groundwater, etc., and this protects the lake from eutrophication. So to conclude, the threat of algae blooms is not to be disregarded. While Lake Natoma and Weber Lake are not under duress currently, this can change very rapidly, and it's imperative that we protect our aquatic environments for both our health and that of the ecosystems that rely on these bodies of water. As for my acknowledgements, firstly, I would like to thank Megan and my mentor Sean for teaching the manuscript writing process and guiding me through this research. I would also like to thank my dad for helping me with the data collection and my mom for helping me find the equipment I needed. Thank you.
Great job. That is an awesome project. And let me get rid of your slides here so we can see everybody. But Sean, did you have any questions?
Yeah, I do have one. So as far as revisiting the lake, what are some things that you would want to look into?
Yeah, so I think with Lake Natoma, that's a specific lake that I would definitely want to go back to. So typically what happens in elongated reservoirs like Lake Natoma is there's a specific area where extremes of high and low turbidity meet, and you can find a pocket of a lot of nutrient content and algae blooms. And I think because of my limitations in collecting this data manually, going back to Lake Natoma and exploring different areas could be valuable, as well as looking at deeper water samples, because these were both pretty clean systems. And so a lot of the nutrients will be sitting at the bottom of the lake, and I had a 12-foot limit.
So potentially looking at some soil or sediment from underneath.
Yeah.
Awesome. I want to hear more about how you designed your sample collection implement, and did you find blueprints for that, or was that all you guys coming up with how to make that?
So that was what I thanked my mom for. We sat around and talked about that for a bit because a lot of the equipment online was really expensive and kind of complicated, and I figured that there was a way that I could build this in a simpler mechanism that would be easier for me to deal with because I was on a paddleboard, which isn't the most stable situation. But yeah, so I went to Lowe's, I got a window cleaner, and it's like a 12-foot pole, so it's extendable, and I can like make it more compact. And then I just added a bottle to the base of it with a cork and a string. So essentially I would stick it in the water, and then I marked it with like foot increments, and once it got to the depth I wanted, I would tug the string, the cork would come out, and you could see the bubbles rising. Once the bubbles stop, then I know it's full. And when you pull it out, the water pressure prevents any excess water from getting in.
Awesome. Very well thought out. I love it. And it seemed to work well to get you some good results.
Yeah, I only had to dive down to get one sample, so otherwise it was fine.
Okay. Sean, do you have other questions?
No, I admire the brilliance in the tools and what you utilized. I mean, that's part of the work, right? When you're out in the field, it's how do you get it done?
Yeah.
So Great job
Thank you.
with that. Mhm.
Awesome. Yeah, fantastic. I love that. And it's awesome that you can go out and collect that data with your created tools. So fantastic job.
Thank you.
All right. Next up, Sean, you've got one more here. I'd love for you to introduce David.
Yeah, it's my joy to introduce David. David has had a keen eye for data and data manipulation, which is kind of explains what his research is. So David used image classification and machine learning for misleading information. I'm not going to give away too many specifics after that. I'll let him explain.
All right. Thank you, Sean. My name is David, and my study was on comparing the effectiveness of image classification models and multimodal large language models in detecting misleading bar charts. Let's first get through some background information. So we all know that we live in the information age. Across the world, there's over 5 billion people who use the internet every day, and on average, they spend over 6 hours on the internet and over 2 hours on social media every day. And as our world has become increasingly digitized, information has become ever more accessible, and also it's become a lot easier to spread information. This survey from Oxford Economics showed that 92% of YouTube users reported that they use YouTube to gather information and knowledge. And this shows just how deeply social media and the internet has become ingrained into our society. People all around the world see social media and the internet as a source of information and news. Unfortunately, these platforms have become plagued with misinformation. I'm sure we all know what misinformation is. It's just information that's objectively incorrect or can be disproven. And in certain situations, the consequences of this misinformation can be severe. For example, in disasters or in politics, or when the misinformation relates to health issues, misinformation can result in pretty severe negative consequences. One survey of YouTube videos relating to COVID-19 during the pandemic found that over 27% of videos contained misinformation, and these videos in total had 62 million views. It just shows how many people actually consumed this misinformation. Also, another survey found that almost 20% of videos presented in the search results on TikTok contained misinformation. And this it makes evident how much misinformation there is on social media, how prevalent it actually is. Currently, of course, social media platforms want to combat misinformation because it has negative impacts on society and also their platform. They employ 3 methods. The first method is simply just algorithmic content moderation. Some platforms like Twitter use simple keyword checks. For example, if you were to tweet derogatory language or something containing a racial slur, it would be flagged and hidden. Other sites also use AI content moderation, which is what we're going to be focusing on today. AI content moderation, the AI content moderation that we see today is primarily made of blocking out violent images that are inappropriate or something similar to that. There's also crowdsourced content moderation, which is basically where a platform uses the user base of the platform to help moderate the content on the platform. The issue with this strategy is that oftentimes relying on the users within a community to be objective, like third-party observers to determine whether or not something is misinformation, is usually these people within a community are unreliable because of how social media algorithms create echo chambers, and everybody in a community will share the same opinions. As a result, this is somewhat less effective. There's also manual review by human workers, and this often works in tandem with crowdsourced content moderation. People can report malicious content or content that violates the guidelines of the platform, and then these employees, these moderators, will just review the content to see if it should be removed from the platform or not. We're going to be focusing on AI content moderation. Specifically, we are going to be looking at how we can fight misleading bar charts. Misleading bar charts are basically bar charts where the proportion between the sizes of the bars is different from the proportion between the values represented by those bars. So what does that mean? Well, if we look at the example bar chart on the right, when you just look at this, you get the idea that if Bush tax cuts expire, the top tax rate will be much larger than it is now. Because when we look at this chart, what we first notice is that the bar on the right is much larger than the bar on the left. But if you pay attention to the values of the bars being represented, these values are not actually that much different. And the difference is not nearly as much as the sizes of the bars would lead you to believe. And this is because the y-axis of this bar chart is truncated. It starts at 34 and it doesn't start at 0. So it ruins the size scaling. The same thing happens on the left side. You can see it's a similar misleading bar chart, and these bar charts just lead to exaggerated differences and possibly misinformed conclusions. Currently, the only approaches to dealing with these bar charts that are often image format or only in image format is through artificial intelligence algorithms. There are two options that we'll tackle in this study: multimodal large language models, which are large language models that can process inputs and generate outputs of multiple types of data, like images and text. That's what we're doing right now. And also image classification models, which process image input and generate, well, technically they generate a number, but that turns into a Boolean if you do some processing. It basically just tells you, is this image misleading or not? Image classification models, in comparison to multimodal large language models, are small, less expensive, faster, and also not as resource intensive, at least in this context where we're using relatively lightweight ones. And they should be theoretically excellent at the task that they are specializing in. Multimodal large language models, on the other hand, are large, costly, slow, and resource intensive. But it's all of these things because it's capable of excellent performance in just general tasks as a result of their large size and the amount of data that they've been trained on. Social media platforms would definitely benefit from more effective automated anti-misinformation methods, and that's what we're going to try to investigate. So how exactly did we train our model, and how did we test our models? Arguably, the most important part of machine learning is collecting data, and good data. We didn't really have good data, but we basically created 3 data sets that we would use to train and test our models. Our first data set is just, it's created by a program that we wrote that would generate misleading and non-misleading bar charts with matplotlib library in Python. Our second data set is composed of non-misleading bar charts that are just downloaded from a publicly available data set. Unfortunately, these bar charts are also generated with matplotlib, but yeah. And the third data set is composed of downloaded images from the internet. These are in various formats and are very different from the ones generated by matplotlib. Here are some examples of bar charts generated. Here are some examples of bar charts in dataset 1. You can see the bar chart on the left is non-misleading. The bar chart on the right is misleading because the y-axis is truncated. These bar charts are varied in size, scale, orientation, data, and colors. Our second data set is similar, except they are varied in orientation, data, colors, and grid layout. And also, there are only non-misleading bar charts in this data set. Our third data set is composed of these completely different and alien bar charts. The features are very different from those in our first 2 data sets. The one on the left is non-misleading, and the one on the right is misleading. These are all scraped from Google Images, and there are not many of them. Because there are not many of these bar charts in dataset 3, we weren't able to find too many on the internet just by searching. We used datasets 1 and 2 to train our image classification model because dataset 3 was unfortunately too small. This means that our model would be specifically trained for the task of recognizing misleading bar charts generated by matplotlib. That means that it might not be able to recognize misleading bar charts generated by other programs because they are presented differently. And here's just the architecture of the neural network we used. It's very simple, elementary. This is just a standard looks like a pyramid, standard design. And moving on to GPT-4o, in order to interface with GPT-4o programmatically, we created this prompt that would first ask GPT-4o to determine whether or not the image of the bar chart we provided is of a misleading bar chart, and then number two, have it give a justification for why the bar chart is misleading or not misleading. And also, finally, we asked GPT-4o to provide its response in JSON format, which is a format that can be easily parsed by pretty much any programming language, and this can be implemented in a program. So yeah. And in total, we had 3 trials to evaluate our machine learning models. Our first trial was composed of bar charts from the first data set. Our second trial was composed of bar charts from our second data set, and so on. All of the bar charts in these data sets are completely new to our image classification model. So our image classification model has not seen these bar charts in training or in evaluation. The first 2 trials would test the ability for our model to perform in its specialized field, and the third trial would test its ability in the generalized task. Now, here are our results. We found that our model performed exceptionally in the first trial, a lot better than GPT-4o in labeling misleading bar charts correctly. They performed both perfectly in the second trial, and GPT-4o far outclassed our model in the third trial, with our model performing worse than just a coin flip. Now, let's just look at some examples from the first trial to see an example of why GPT-4o performed a lot worse than our model in recognizing misleading bar charts. So here we have an example of a misleading bar chart. The y-axis is truncated at 0, it doesn't start at 0, it probably starts at something around 2. And GPT-4o labeled this bar chart as non-misleading. Let's see its reasoning. It says that the bar chart is not misleading because the y-axis starts from 0. And that's just not true. So this just shows that the visual understanding capabilities of GPT-4o are just not there. They're not perfect. They're pretty flawed. And as a result, it sometimes gets something simple like this wrong. Our model, on the other hand, is almost 100% confident that this bar chart is misleading because, well, yes, this is demonstrating its excellence in the field, the task that it specializes in. On the other hand, this is an example from the third trial. GPT-4o correctly recognizes that this bar chart is misleading. It says that the bar chart is misleading because the y-axis does not start from 0, but instead starts from 79,000. And yes, that means the y-axis is truncated, and so this bar chart is misleading. Our model, on the other hand, says that this bar chart our model, on the other hand, is 100% confident, almost 100% confident that this bar chart is not misleading, showing that our model just is confused. It does not understand this input, and honestly, I wouldn't blame it because there weren't any images with this type of blue background color in its training data, so it does not know how to approach this data. What conclusions and insights can we draw from our results? Well, we found that our image classification model did achieve a greater accuracy than GPT-4o on datasets within the scope of the task it's specialized for. In fact, it achieved pretty high accuracies. I think it was 98.3%. We also found that even though GPT-4o is more generalized, it demonstrated significant variance in its accuracy between trials 1 and 3. That has to suggest that there is a difference between trials 1 and 3. And I think the possible difference is that trial 3 is like publicly available data, and it might have actually been part of GPT-4o's training data, while trial 1 is completely new data. So GPT-4o would not be as familiar with the data presented in trial 1 as in trial 3, because we know like these companies that like OpenAI, they scrape the entire internet for training data, and it's quite possible that the images in the third trial were part of its training data. We've proven the ability of image classification models to recognize misleading statistical graphs within the scope that they are specialized for. So As a result, further research should work on expanding that scope, enhancing the ability of these models to perform in more general tasks. Future research can work on this by creating larger and higher quality data sets with real data, combined with some synthetic data if necessary, and employing some data augmentation techniques. Future research can also conduct more fine-tuning of the model architecture to perfect it, and also perhaps employ different approaches, for example, ensemble learning, transfer learning, etc. As for what social media platforms should do, they should employ a multi-pronged approach for misinformation detection, where they utilize all tools they have at their disposal. Depending on research constraints and the characteristics of information being posted onto their platform, such as the volume, developers should leverage these specialized image classification models and also generalized and also multimodal large language models that are capable of generalized tasks. For example, a social media site that has lower information volume but wants the information to be higher quality might employ a multimodal large language model, while a site that has a large information volume and does not have the ability to employ multimodal large language models because of the resources needed should just use these smaller specialized models. These algorithms should also be integrated with other non-algorithmic anti-misinformation measures. So, for example, what I said before, if a platform has a lot of information volume and it can't use multimodal large language models to filter its content, it might want to use these specialized models to label or just suggest that to post is possibly misleading, and then combine that with crowdsourced content moderation to have the users of the platform vote on whether or not the post is actually misleading. So thank you all for listening. I hope you learned a lot about misleading bar charts.
I learned a ton from meeting with you the other day in your practice and talking to you in class, and from this final version of your presentation. Incredible. I have one question for you, which is actually not about misleading bar charts, but if you were going to do something similar with another kind of chart or another kind of, misleading graphic, what would you change?
I mean, I think the main issue for me right now is just data. So I would just be working on creating larger and higher quality data sets, because the issue with data is if you just go on Google Images and you search for misleading bar charts, you won't get many, and half of the misleading bar charts aren't actually misleading. So if you were to just programmatically scrape all of those images, you would still need somebody to manually go through those images and label them. And especially when it comes to making larger data sets, that would take tremendous work. So yeah, I know large companies just hire, they outsource their labor, but I personally do not have that capability.
Of course. Sean, do you have a question?
Yeah, I kind of have a philosophical question. So I was thinking about this, David, and what advice would you give to somebody who wants to know, how, right, they're on, like you discussed, they're using the internet, they're utilizing their social media. What would you recommend to do to kind of avoid or just try to, I guess, truncate your view or vision of some of these misleading images?
Well,
or receiving your news, right?
I mean, on social media, it's hard to tell, because it's often not just a matter of misleading charts, it's just charts with wholly incorrect information. So that can't really be detected by something like a simple image classification model. You need a multimodal large language model might be able to detect incorrect information because they have something similar to an understanding of the world. But I would say you just have to diversify the content that you see on social media. So you should visit communities that share basically all perspectives. And generally, if you look at what everybody is saying, you can get an idea of what's true and what's not.
Awesome. I've got one question here from our audience, and this one's about catching different graphical communication strategies.
So I was thinking maybe you could first implement a model to just categorize or classify what type of graph. So maybe first you could have a model that would just look at an image and say, is it a graph or not? And then a second model that would just classify what type of chart it is. And then a third, and then in the third layer, you would just run your specialized model to detect, to label the graph, like whether or not it's misleading. Yeah.
Awesome.
So yeah, that would be a lot less resource intensive. I'd say during my training process for my model, which I performed on my own computer, I probably used like 20 watt hour, I don't know, maybe 30 watt hours of electricity. And if I look at electricity prices, that's like less than a cent. While doing just evaluating my graphs on GPT-4o using OpenAI, that cost me like $1, I think. So yeah.
Awesome. Well, great work. Very impressive coming up with the whole thing. And if you have a chance, there's a couple more questions in the chat that you could answer via chat, but we are going to move along. But fantastic work. Thank you so much.
Thank you, Sean, as well. Bye.
Of course.
All right. And so next I'm going to bring up Jesse, who is going to introduce our duo for the night, Lauren and Easton.
Hi, hope you all are doing well. Yes, I'm proud to present Lauren and Easton and their research project, which is the influence of water quality on mollusk abundance. This is a study of the South Bay region of San Francisco Bay, California. And Lauren is a student at Aragon High School near the same location as the study, and is Easton as well. He is a student at Mountain View High School. And both of them conducted an outstanding research project. They worked very well collaborating on their analysis and conducting this research and developing the statistical analysis in the presentation. And I will turn it over to them. I'm super proud of them.
Awesome. Take it away, guys.
Hi, so yes, I'm Lauren Tazbaz, and this is what our presentation is on, the influence of water quality on mollusk abundance in the San Francisco Bay. So the San Francisco Bay covers an area of approximately 1,400 square kilometers and borders major cities like San Francisco, San Jose, and Oakland. It hosts over 500 species of fish, birds, mammals, and shellfish. Our research took place in the South Bay region, which is where that purple arrow is pointing to, which is in the San Jose area. So the San Francisco Bay is home to 3 notable native shellfish species, the Olympia oysters, California mussel, and hybridized bay mussels. These mollusks provide crucial ecosystem services, including water filtration, so removing nutrients from the water, improving water quality, habitat creation, so creating habitats for other aquatic organisms, and wave attenuation. This helps in coastal stabilization and protects aquatic organisms from other environmental forces.
Threats to native mollusks. Native mollusks have a low range of tolerance to habitat threats, such as predation, habitat competition, sedimentation, low salinity, pollution, and water contamination. Some invasive species to the San Francisco Bay Area include the Asian clam, the Japanese littleneck, and the Asian saumeel. Invasive or non-native species exhibit a larger range of tolerance, and the wide range of water quality in the San Francisco Bay makes it easier for invasive species to outcompete the native mollusks, leading to large populations of non-native and invasive species. So our research question was, which factors of water quality affect both native and non-native mollusk populations? Now you may ask, why is this research important? Knowing this will help us better understand how to restore native species and protect them, and the removal of keystone species, such as the native ones like the Olympia oyster, can snowball negative effects. Our hypothesis was that areas with higher pollution levels, such as those near urban centers, will have significantly smaller and less healthy native mollusk populations, and a larger number of invasive mollusk species.
So this map displays our study area. The red circle is where we conducted our research in the lower South Bay region. These yellow dots are water quality stations that we utilize in our data, and we focused on water quality stations 29 to 36. This is a closer look at that area. The blue pinpoints represent the water quality stations. That blue stream in the middle represents mollusk abundance. So as you can see, the abundance of mollusks decreases as the bay gets more narrow and closer to urban and industrialized areas. If you look on the map, you can see a little heart and an arrow. The heart represents where I live, and the arrow points down to where Easton lives. And this represents how this study and research impacts our life as it really is happening in our own backyards.
Research method plan. So the first step in our plan was to acquire data. The second step was to prepare and analyze the data, and then the third step was to model the data. So to acquire the data, we first utilized two data sets from previous studies. One was a 2019 water quality data set from the USGS. The other was also from 2019, a data set obtained from USGS on mollusk populations in various areas of the bay. Then, to prepare and analyze the data, we mapped the data points in ArcGIS Pro and joined information from the nearest water quality testing area to each mollusk population area. We then analyzed the water quality factors in areas with high and low mollusk populations to identify significant influences on population health.
So we started with normality testing. We saw that the data was not normally distributed, so we applied a square root transformation to the data. But after we did this, the transformed data also did not meet the normality assumption. So we moved on to outlier detection. We used box plots and a Grubbs test to detect outliers in the mollusk abundance data, and then we excluded the species Asian clam. There's a very large abundance of the Asian clam species, so by excluding it, it condensed our data and made it easier to understand. So these data figures represent mollusk abundance. The one on the left represents total abundance of mollusk species. That blue horizontal line represents the mean, and the orange line represents the median. And on the right is another form of the data on the left. It's a histogram, and again, the blue line represents the mean. So then we move on to correlation analysis. We visualize correlations using core plot function in R. The correlation analysis between water quality variables was performed to identify potential relationships between different environmental factors. Then we use generalized linear models, or GLMs, to analyze the relationship between mollusk abundance and species in R. Then we identified significant factors. Lastly, we did our water quality analysis to explore the relationships between water quality variables and mollusk abundance.
So here are our results for the water quality. As you can see, figure 4 shows the relationship between oxygen concentrations and temperature. There is a significant negative relationship between water temperature and dissolved oxygen levels. Lower oxygen levels can lead to hypoxia, decreasing native mollusk populations. Additionally, a weak but significant negative relationship was found between calculated SPM and dissolved oxygen. The graph is not shown. Figure 5 shows the relationship between oxygen levels and water quality stations. Station 33 has the lowest oxygen level because it is nearest to urban industrialized areas. And as you can see here, figure 6 shows the relationship between calculated suspended particulate matter and the water quality stations. As you can see, there is significant spatial variation, and again, station 33 has the highest levels of calculated SPM because it is nearest to urban areas. So the effects on mollusks. All in all, factors that reduce water quality include higher levels of SPM, lower levels of oxygen concentration, lower levels of salinity, and lastly, higher levels of discrete chlorophyll A, which leads to higher levels of nutrients such as ammonium and phosphate. This reduction in water quality creates favorable conditions for invasive species since they've mostly adapted to it, while reducing habitat quality for native species. Figure 7 here shows the abundance plots per species excluding the Asian clam. Most of these species are invasive because many native species have already practically died out due to the poor water quality.
So in our discussion, we highlighted that the study of water quality parameters provides a very comprehensive understanding of the aquatic environment, which directly influences the abundance and distribution of mollusk species. Our research proved our hypothesis by displaying that water quality factors in places closer to urban industrialized areas were worse and therefore less suitable for native mollusks. However, since invasive species are able to adapt and flourish in areas with poorer water quality, there is a much larger population of invasive species, proving our hypothesis as well.
So what does this all mean? It means a loss of native species, an increase in invasive species, a loss of native habitat, which ultimately leads to even poorer water quality.
However, it is not all doom and gloom. Our research shows that there are ways for effective management. Restoration efforts should focus on improving water quality, reducing pollution, and restoring natural habitat conditions to support resurgence of native mollusks because, as we know, they are very important to their ecosystems. Lastly, we'd just like to thank everyone who made this presentation possible, Megan, Joyce, and Sean Wesley for helping us with our presentation, our manuscript, and teaching us classes, our classmates who helped us in the peer reviewing process, and Karina Sanchez for reviewing our manuscript and leaving very helpful comments. Thank you.
Great job, you two. That was wonderful. I'm going to bring Jesse back up here as well. And Jesse, did you have any questions for these guys?
Yes, first, that was an outstanding presentation, Lauren and Easton. Terrific job explaining the correlation between the spatial analysis that was conducted and the changes in the water quality and the urbanized areas. And how the water quality and poorer water quality leads to that increased abundance of non-native species, which are more adapted to, or I should say, more tolerant of reduced water quality. I do have a multi-part question for both of you. Considering that the most recent mollusk species abundance data that we had was from 2019, which was the last time that data was collected, would you change any data collection methods, or perhaps would you change the location of the study to focus on native species, or would you continue focusing the study on how the abundance of those non-native and invasive species are changing in relation to that water quality we observed?
So I can answer that first part. I don't think I would change any of our data collection methods. They were very helpful in giving us clear data. I think, though, however, I might want to change the location to other areas of the bay since there were almost pretty much no native mollusk species located in the lower South Bay. I definitely want to go up north more, maybe areas even that are way less industrialized, so that we can see really the impact of aquatic pollution on the mollusk abundance.
Excellent. Thank you, Lauren.
Yeah, and obviously it wasn't like a preference for us to focus on these non-native and invasive species. Unfortunately, there really aren't that many Olympia oysters or other native species left in the bay, and it's very hard to find them, and any information on them would be pretty much non-existent in the lower South Bay. So obviously, yes, we would move our study up to figure out more about the Olympia oysters, and hopefully as time goes on, the restoration efforts succeed, and hopefully there will be Olympia oysters will be reintroduced back into the lower South Bay, and then we could really do a nice research project with the information there.
Great. Thank you, Easton.
Awesome. We have a question from the audience for you guys. Can this research translate to research on invasive species in other bodies of water, specifically in temperate regions, but as invasive species kind of continue their move? What do you think?
Want to take this one?
Yeah. Wait, do you want to answer, or should I?
You can go ahead if you want.
Okay. Yeah, I think this research could work on other places. But I think it would be good if to conduct this research just in other areas of the bay just to check. But I think generally the results we came up would be the results that would come up in other areas of the world.
Yeah, I agree with Ethan. Just to add on a little bit, invasive species really do have similar impacts, and wherever they are, they take out native species. And if the native species serve very important ecosystem services to their aquatic environment, then it leads to poor water quality and poorer effects on other aquatic organisms. So I do think that this can translate to many other areas.
Awesome. And just a quick question for both of you. You can pick either the most surprising challenge that you encountered with your research, or something that you really enjoyed that maybe you didn't even think you were going to get to do?
So, for me, the biggest and most surprising challenge was our initial plan was to figure out which species were native and which species were invasive, and then we would conduct a t-test to compare the two and what water qualities they were adapted to. But it came as a big surprise when every single one of the species on that list turned out to be invasive, and it was really saddening to hear that the native species their numbers were just negligible. There were barely any of them, and definitely not in the lower South Bay. So that was a challenge that we had to overcome, and it was also surprising and saddening as well.
Yeah, I totally agree with Easton. Something that I did enjoy doing, or like enjoyed to see, was when we created our data figures. It was just very cool to see like everything fall into place and almost very nicely prove our hypothesis. And though it's sad our hypothesis was proven, it was nice to see that, like, oh, we were on the right track with our data figures.
Yeah, awesome. That was something that we heard in from some of the students in the earlier presentation session as well, how nicely, doing your stats and looking at that analysis kind of wraps everything up, and just seeing how some of those numbers, can mean, large numbers can mean one thing, small numbers can mean another thing, and kind of putting that all together to answer your question. So great job, both of you. And thank you so much. We are going to now bring up Warren, who is going to introduce Oscar.
Hello. So I had the privilege of mentoring Oscar for this summer's research experience. His project was focused on middle distance track and field events, so very timely. And he brought a lot of enthusiasm and sort of personal experience to the research project. And he worked hard on some novel questions and put together a pretty robust data set to look at them. So without any further ado, take it away, Oscar.
Thank you. Okay, so this summer I researched the improvement rates and career trajectory for track and field middle distance high school and college athletes. My name is Oscar Wettenwerk, and I attend Menlo Atherton High School, and I play soccer and I run track. Okay, so the introduction. Through this project, we, along with other people, we investigated the relationship between athlete improvement rates and career trajectory. And in this study, I will address two different questions. And those questions are, how did the high school improvement rates of the lower tier of top-ranked college athletes compare to the high school improvement rates of the top tier of top-ranked college athletes? So in this question, we're basically comparing two groups, which are the top-tier ranked college athletes and the lower tier ranked college athletes, and we're comparing their improvement in high school. Second question is, how does improvement in high school years correlate to improvement in college years? So in this question, we're seeing if there's any correlation between the improvement in high school versus the improvement in college. Not much is known about this topic, and because of that, I thought it was an interesting topic to investigate. As a high school runner myself, I want to have a deeper understanding and learn more and understand more about these topics. Introduction part 2. So now I'm going to talk about middle versus long distance races, or versus short distance races. Middle distance races are the 800, the 1500, and the 1600, versus short distances are the 100 meter, the 200 meter, and the 400 meter. In a middle distance race, an athlete tries to max out their aerobic capacity by the end of the race so that they know that they couldn't have gone any harder and they got the fastest time possible. Versus in a short distance race, an athlete attempts to accelerate to around max speed, depending on the race, and then they try to hold that speed till the end of the race. And there's a difference between high school and college, because in high school, a lot of runners are multi-athletes. So they're not only focusing on one sport, but they're focusing on multiple sports, which can lead to injuries, and it just makes it a little bit harder. Versus in college, it is likely that an athlete is most likely focused on track and field and cross country, just solely running. My hypothesis is that I believe that the improvement rates of the bottom tier college athletes are higher. And I think the reason I think this is because I think that the top tier athletes, they developed earlier, and they've been running since they were young. So I think for that reason, the bottom tier athletes are going to have improved more. And I also think that the high school improvement rate has little to no correlation with college improvement rate. Okay, now on to the methods. The data that I collected for this project is from the athletic.net plus website, and the athletes, which is a subscription on athletic.net. Basically what athletic.net is it's almost all of the US runners. There's events, it's times, it's results, it's multiple different things. But the athletes that I looked at are the 2024 senior males in the US with the best 1500 meter times who raced D1 NCAA college track and field. I found data across all 4 years of college for high school and college. And I ended up with a random sample of 50 runners, 25 from the lower tier and 25 from the top tier. The sample was randomized with the use of random.org, which is a website which basically randomizes the numbers that you input into the numbers box. Methods part 2. So this is a 5-step process of how I gathered my data. So I created a ranking based on criteria for athlete selection. And then I went down the randomized numbers to find athletes that can be included. And then I copied 3 or 4 years of college PRs. And then I had to find the high school profile for that athlete. If the college athlete doesn't have an integrated high school account, then I had to search for their high school by name. And then if several people have the same name, you have to research to figure out, which high school did this person go to? And a lot of them didn't have their high school data on athletic.net, so that made it a little bit tricky. And then the last step was to copy 3 or 4 years of high school PRs. So what they got there from their first year to their last year in high school. Now I'm going to talk about my results. So as you can see on the left here, we have our graph. And for the bottom tier college athletes, they had an average improvement of 6.12%. So that is the orange bar that you see there. And the standard deviation was 3.647. For the top tier college athletes, they improved 8.55%, which is the blue bar there. And the standard deviation for them was 3.655. And the p-test, or the t-test, to compare the two groups, we got a p-value of 0.036, which is below 0.05, meaning that the results are significant. And this little bar that you see on top of the blue and orange bars, that is the standard deviation. Okay, results part 2. So for the other graph, we used linear regression to test strength and direction of correlation, if any, and we did college versus high school improvement rates. So we got an R-squared value of 0.019, which is very low, meaning that the correlation is very weak, and a p-value of 0.335, meaning that the correlation is not significant. Okay, now on to the discussion. So the top tier athletes improved more than the lower tier athletes, which led to them running a faster time by their senior year in college. It can be inferred that the more an athlete improves in high school, the greater the chance they have of achieving success in college. And for the second experiment, we can confidently say that there is no significant correlation between the athlete's high school improvement rates and their college improvement rates. So if an athlete improves a lot in high school, it does not mean that they're going to improve a lot in college. There could be a correlation, but even then it would be very weak, not much significance. A possible bias is that perhaps not all seniors in the US are actually on athletic.net. This could be for many reasons, but yeah. A possible future experiment would be to look at this from a non-US perspective. So athletic.net is mainly US athletes, so there's not many athletes from other countries. So it'd be nice to look at other countries. But it would also be nice to look at the female track and field athletes, or to look at D3 and D2 track and field teams, because in this project, I only looked at D1 athletes. Okay, acknowledgements. I just want to say thank you to Warren Saber, my mentor, my parents, Megan Joyce, and Sean Wesley for providing me with support and help throughout the creation of this project. They all supported me in some sort of way, whether it was helping me with data collection, editing, and giving suggestions, providing me providing me with the supplies needed to complete this project, or helping explain concepts I didn't understand, or helping me practice presenting my project. Thank you for listening.
Great job, Oscar. That is fantastic. And Warren, you're right, very timely. Do you have any questions for Oscar?
I do. First of all, this is kind of a two-parter. Why did you choose to focus on middle distance events exclusively? And do you think the patterns that you saw here would hold true for shorter sprinting events or longer, more pure endurance events?
Okay, so I decided to work on or to look at middle distance runners because I personally run the 800 meter and the mile, which are middle distance races. And also I just think those are very interesting races. So yeah. And then I think that the results, I think that they would have been a little bit different for short distance races. I think that there still wouldn't have been any correlation between the high school and college improvement rates. But I think that there definitely could be a difference between, or a bigger difference between the top tier college athletes and the lower tier college athletes.
Something I noticed in your data is that the range of improvement in high school is about 1.5 to 2 times as wide as the range of improvement for athletes once they get to college. Do you have any thoughts on like why there's so much of a wider range in high school as compared to college?
Sorry, can you say that again? I was good.
So in your second question, when you were looking at high school improvement rates versus college improvement rates, just looking at the way that data was distributed, you have a very wide range of improvement in high school, maybe, between 0, I think a little below 0 and like 18%.
Yeah.
And then a narrower range in college, only up to about 10% with, a few athletes with negative improvement in college. I was just wondering, do you have any idea why you might see a wider range of improvement in high school as compared to college? Why aren't people, continuing to hit those, 15, 18% improvement rates in college?
Yeah. Okay. So in high school, I believe they improve more because of obviously puberty, and I think it's that some of them even start running then, so there's a lot of room for improvement. And yeah, obviously puberty and growing and development, and some athletes even peak in high school, right? They get really fast in high school, and then in college they don't improve much. So I believe that's the reason why the high school athletes improve more. Even though college athletes, they mainly focus on running in college, they've likely already reached like a very high level, so it's hard to get faster. And also it's a lot harder to get faster when you're at such a high level and when you've been racing for so long. Every second counts as a lot. So yeah.
And then I just have one more that sort of keys off that a little bit. Did you see any like weird, any anomalies in your data? I'll say, because one thing that occurs to me is that behavior and attitudes can affect race time a lot. And I don't know if you see something where people aren't improving for a long time and then they get really serious and improve a lot. Did you see anything weird in your data that you felt like you had to account for?
Yeah, for sure. There was one case, or there was a lot of cases, where athletes in high school, their senior year, they got a lot slower and I think the reason because of this is that colleges really look at athletes in their junior year. And so that's when they try to race their fastest. And then I think in college years, they might have less classes and they don't need to improve as much. Like the colleges have already looked at them and studied them. So they don't decide to focus on track as much. They lower their focus on that, and this leads to them just running slower in their senior year.
Okay.
Awesome. Yeah, I had not really a question. I feel like you answered it, but in some sports, I feel like there's a like growth and then a plateau for a while, and then you hit a spurt of growth again. And so just kind of thinking about those trends in your data, and if running, shows some of those kinds of improvement as well, you could potentially look at post-college athletes who, go on to make the US team later in life or something like that in a future study.
Yeah, for sure.
Awesome. Well, great work. Fantastic, super interesting, and as we've seen, very different than some of our other topics so far, which is super fun. Awesome. All right. And next, I'd love to bring up Haley, who is going to introduce Lucy.
Hello. So Lucy has been so awesome to mentor this summer with her project. She has put in a lot of hard work in collecting samples from about 6 different lakes/reservoirs. So I'm really excited for you all to see what she found. So without further ado.
Amazing. Take it away, Lucy.
So my name is Lucy. I'm a junior at Santa Cruz High School, and this summer I focused my research on assessing microplastic levels among various lakes in the Truckee River system. I started looking into this topic because I knew I was doing this program and I was really excited, but I wasn't sure what to focus on. And my mom brought up the idea to look at microplastics in the lakes. And when I was looking into that, I hadn't realized how much fresh water is at risk. So I learned all these interesting things about freshwater, including that only 3% of Earth's water is freshwater, but freshwater ecosystems are extremely diverse, and more than 100,000 species, including humans, rely on freshwater. But unfortunately, there are lots of water shortages in the world, and 2.7 billion people experience water scarcity. Humans have adapted to use natural resources, including freshwater bodies, in many ways. This includes dams, irrigation, and agriculture, and recreational uses like fishing and paddleboarding, and I love to go paddleboarding with my dog. So, hold on. Some of the most prominent threats to our freshwater ecosystems are pollution, overfishing, dams, and diverting water sources. And there are lots of ways to combat this, but those measures aren't always put in place. And since the 1760 Industrial Revolution, humans have created 8.3 billion tons of plastic. And one of the most famous consequences is the Pacific Garbage Patch, which is a floating island of plastic larger than the state of Texas. And plastic pollution has been known to kill more than 100,000 marine mammals per year. So, microplastics are a relatively recent discovery. And according to National Geographic, in 2021, there were 24.2 trillion microplastics in the upper oceans. Microplastics have been found in the body, causing cancer and infertility. And there have also been studies done with mice where they're also causing reproductive harm to the mice, both male and female. Most microplastic is so small that it can't be seen by the human eye, but it is very dangerous. And most microplastics are smaller than the top of a pencil eraser. And under UV light, microplastics have fluorescent properties. So you can see on the bottom right, that is an example of under the microscope, the microplastics reflecting. So my study focused on the broader Truckee-Tahoe area, and it was a very unique context for further investigating the relationship between human activity and microplastic pollution, because there are high levels of recreational water activity and varying amounts of urbanization across the bodies of water. There have been several important studies of microplastics, but there's only limited research of more specific lakes and reservoirs in the area, and most of the research is focused on Lake Tahoe, which is one of the largest lakes in the area. My main objective and my focus question was how many microplastics had infiltrated other local lakes and reservoirs around the Truckee North Tahoe region, and what patterns and variations are found at various locations with different levels of human use and urbanization. So I hypothesized that local lakes with higher foot traffic and public use would exhibit increased concentrations of microplastics on their shores, while remote lakes protected from human contact would have little to no microplastic contamination. So for my methods, I selected 5 sample sites, different lakes and reservoirs with varying levels of urbanization. So like one of the earlier projects, I've studied at Weber Lake, but then also Donner Lake, Independence Lake, Boca Reservoir, Stampede Reservoir, and Prosser Reservoir. I took samples on the north, south, east, and west shores, 3 millimeters in with a grab bottle. Sometimes I hike down and sometimes I paddleboard, but I made it work. And one time I was like sliding down a hill trying to get in there, but I did it. And then after that, I took 3 subsamples from each of the shores and I looked at them under a microscope using a UV flashlight that I taped to a pitcher so I could shine it on my station. And then from there, I looked at the slides and counted all the microplastic particles. So this is an example of what I was looking at under the microscope. So on the right, there is a piece of fishing line that is reacting with the UV light in the second photo. And then, oh, that's not on the right. That's on the, no, that is on the right. I don't know what I'm saying. This is the left. On the left side, we have I believe it is a wing of some type of bug, but then you can see how there are little chunks of plastic that were also on the sample. And you can see how you can tell which one's which. So my results were that there was a greater microplastic presence in lakes with higher visitor numbers and recreational use, like Donner Lake. So my hypothesis was supported, and the local lakes with higher foot traffic had increased concentrations of microplastics on their shores, and the remote lakes, like Independence Lake, had a little less microplastic contamination, but they still had some. So this was the summary of my findings, the concentration per sample, and the p-value was less than 0.01, which indicated that my results were significant. Okay. So if I did this experiment again and I had less of a time restriction, I would definitely go take more samples on different days, and I would do more subsamples from the samples. And I also believe that if anyone else did this project, they should do it during the different seasons and look at how tourism affects microplastics in the water. And I just thought something really interesting was that the other day I went to Starbucks and they switched from plastic cups to paper cups, and they were trying to be more eco-friendly, but Starbucks cups were also found to put polyethylene and polypropylene microplastics into your water, which is not good for you. And I would just like to send my gratitude to the people who made this project possible. My mom was a constant source of inspiration and encouragement, and she was always there to help me. And my dad, he was always there with me at the lakes. We went together on a bunch of road trips, and it was really fun getting to spend time with him that summer. And of course, my mentor, Haley, I couldn't have done it without her. I was definitely confused on how to do a lot of things, and she was super helpful. And thank you, everyone, for watching my presentation.
Awesome job. That is a lot of work to get to all of those sites and collect all of that data.
Yes, it was definitely, but it was a fun way to spend my summer.
Awesome. Haley, do you have any questions for Lucy?
I do, actually. So you were able to experience what it's like to conduct field work and do a lab analysis. So just out of curiosity, would you say one is more difficult than the other, or what would you say after experiencing both?
It depends. I feel like the lab work was definitely more tedious, but the field work also had its own challenges, especially going to all the different sites, because some of the lakes were super remote and didn't really have good access to all of the shores. But I think there's also fun parts of both. So you just gotta grind through looking at all the samples under the microscope.
Okay. And then I have one more question too. So, microplastics definitely is kind of a newer, like, hot topic. And I know just from your study, it sent me down a rabbit hole of just researching everything that's contaminating us now. Has it changed your perspective on a lot of things, just about like lifestyle and, like you mentioned, the Starbucks cups? I mean, do you see kind of life through a different lens now that you've kind of looked at it at a micro level?
Yeah, I definitely feel like I'll be more conscious making choices about like buying drinks that come in plastic bottles and food that comes in plastic packaging, and also just using less plastic in general and trying to contribute less to microplastics and like all over. Yeah.
Okay.
Really interesting. I had a question kind of along the lines of your answer to your first question. Did you think about marking some kind of observational category of like how trafficked or how much access there was on each side of the lakes? I figure some of the places you went to are like on a road or there's a beach or some kind of access, and then you said some of them you were like sliding down the hill trying to get there without a trail.
Yeah. I didn't do that at the time, but looking back on it, I definitely should have.
Could be interesting, and then
Yeah, that could be an interesting factor to add to the experiment.
Yeah, and then my other question was just how much visible, more macro plastic did you see while you were out there at all of the lakes? Could you see it just anecdotally a difference in the
Yeah.
lakes?
I mean, sometimes when you looked at the water, you could see bigger particles floating on top. But then also when I was looking more closely at the samples, like some of the pieces were pretty big. And like obviously a little orange thing is not like from Mother Nature. So it was definitely, but I feel like I wouldn't have been aware of it or seen it as much if I hadn't been doing this project.
Awesome. Well, yeah, it sounds like you had a really varied experience and had some fun exploring too. So you got out and got to some new places, which is always exciting.
It was very exciting.
Awesome. Great job. And yeah, and Haley, I am now going to have you introduce Ishana.
Okay. So it has been a privilege working with Ishana as well. Her dedication and enthusiasm never wavered once through any obstacles faced this summer. That really allowed her to produce a really, really cool project. And I'm excited for you all to see what she did.
All right, take it away.
Thank you so much, Haley. Hello, everyone. My name is Ishana, and I am presenting my project, which tested the effect of varying salinity levels on the diversity and the abundance of lotic freshwater benthic invertebrates. And the term lotic refers to moving or flowing water bodies. So freshwater ecosystems are defined as water bodies with low dissolved salt and total dissolved solid concentrations, and they are important in several ways. For example, they provide us with multiple ecosystem services, including water supplies, flood protection, food, raw materials, recreational services like fishing, energy, transportation, maintenance of high water quality, and finally, aesthetic and educational benefits. They also mitigate climate change by acting as carbon sinks. Finally, they host tremendous biodiversity, which is vital to ecosystem resilience as well as human survival and well-being. Anthropogenic or human-induced salinization of these freshwater systems is a major threat to them. Freshwater salinization is occurring due to both natural causes such as climate, topography, and vegetation, and anthropogenic causes such as agriculture and pasture, resource extraction, the use of salts for de-icing roads, and urban and industrial wastewater contamination. And climate and land use changes also exacerbate this phenomenon. Aquatic organisms' ability to adapt to these changing salinity levels might be insufficient to protect against the salinization, disrupting ecosystems and negatively impacting human well-being. Benthic invertebrates are invertebrates including insect larvae, crustaceans, worms, mollusks, and mites that live on, in, or near the bottom substrates of water bodies. They serve as a food source for several species, including fish and crustacean species commonly used for commercial or recreational purposes. And this role helps them preserve biodiversity and therefore ecosystem function. Benthic invertebrates are also commonly used as indicator species and maintain ecosystems by enhancing nutrient cycling and aerating sediment layers. And overall, these activities and other biological interactions create diverse niches that increase the biodiversity of freshwater systems. Studies on Slindy's effect on benthic invertebrates are often conflicting, but understanding this relationship is essential to understanding the impacts of freshwater salinization on ecosystem functioning and human well-being. As a result, in this project, we examined the effect of varying levels of salinity on the abundance and diversity of benthic invertebrates in 3 California rivers, and we hypothesized that increasing salinity would lead to a decrease in their diversity and abundance. We used data from 2 data packages from the Environmental Data Initiative, discrete water quality monitoring in the Sacramento-San Joaquin Bay Delta, and benthic invertebrate monitoring in the Sacramento-San Joaquin Bay Delta. And just as a notice, specific conductance is often used as a measure of salinity, and so was used for this purpose in this study. We filtered our data to contain only years 2020 through 2023 to both facilitate efficient analysis and select for more accurate and relevant data. And we selected 3 sites. Sites were selected if they were low-tick freshwater bodies located in Northern California within 25 miles of Suisun Bay, and if they had data available in both data sets. Due to irrigation and water extraction, several low-tick freshwater bodies in Northern California face salinization, and in addition, the effects of freshwater salinization from sea level rise could be more easily observed closer to the bay. And these are the 3 sites I chose, and they're labeled on the map as well. Next, we filtered the data to eliminate data with unknown family classifications because this category could contain multiple benthic invertebrate families or families that were already documented at a specific site and skew our results. We matched monthly salinity data from our 3 sites with the invertebrate data from the same sites and months, next creating pivot tables from this monthly invertebrate data to calculate abundance, family richness, and Shannon index values. And finally, we graphed our data points and analyzed them with linear regressions. The linear regression in Figure 1 that tested average monthly specific conductance against monthly Shannon index values returned a p-value of 0.02 and an r-squared of 0.0446. Although statistically significant with a p-value less than 0.05, the relationship between the 2 variables is only weakly positive, since the variance in specific conductance only explains about 4.46% of the variance in monthly Shannon index values as indicated by the r-squared value. The linear regression in Figure 2 that tested average monthly specific conductance against monthly family richness values returned a p-value of 0.03 and r-squared of 0.037. Again, this relationship is statistically significant, but only weakly positive. Finally, the linear regression in Figure 3 that tested average monthly specific conductance against monthly abundance values returned a p-value of 0.01 and an r-squared of 0.0548. Again, this relationship is statistically significant, but only weakly positive. To repeat our hypothesis, we believe that increasing salinity would lead to a decrease in the diversity and abundance of low-tick freshwater benthic invertebrates, and our hypothesis was not supported as there were significant positive correlations between salinity and benthic invertebrate diversity and abundance. This could be explained with ecological interactions. These include a decrease in predators, an increase in prey, the death of salt-intolerant invertebrates, creating more niches for salt-tolerant ones, and factors that could have led to an increase in abundance. And other environmental conditions, such as fluctuations in factors such as pH, dissolved oxygen levels, and temperature, could also have contributed to our results. There were a few limitations to this study. For one, only 3 sites across 4 years were used, and this may not be expansive enough to discern the true relationships between salinity and benthic invertebrate diversity and abundance. Also, we excluded the unknown category of benthic invertebrate families from our study, which means that we underestimated diversity in some cases and almost always underestimated abundance as well. Future research should examine the effects of multiple environmental conditions and their interactions on the diversity and abundance of benthic invertebrates to get a more holistic picture of what conditions really impact these organisms. And future analysis can also compare groupings of the same 3 to 4 months at a time to account for seasonal variation in invertebrate presence and number due to their life cycles. I would like to thank my mentor, Haley, for her guidance in completing my project and my manuscript. And I would also like to thank Megan, Sean, and the other staff at the Headwaters Science Institute for their support. Finally, I would like to thank Morgan Batty, Sarah Perry, and Elizabeth Wells for creating and making available the data they collected in the Environmental Data Initiative's data portal. And thank you all so much for listening.
Awesome. Great job. I have a question to kick us off, and I know that you had a couple different questions that you came in thinking you wanted to study, and you started going in a certain direction, and this is where we ended up. But I wanted to ask what you've learned about the process of research through all of your different twists and turns.
What I've really learned is to be perseverant and persistent and just kind of keep trying to find a way that you can make it work out. I still ended up studying the kind of my general area of interest for this project, and so I'm really glad that I was able to do that despite the challenges I faced. So I'd say that was the main thing I learned.
Awesome. Yeah. And you did a fantastic job. This is such a good data set, and it really is looking at your question, even if it's not even remotely what you thought you were going to be doing at the beginning. Haley, do you have any questions?
Yeah, that was actually going to be my question too. Just, the challenges you face and how you overcame that. But I mean, in my experience, it just seems to be, your enthusiasm and motivation the whole time. So you kind of answered that question already. But more of a specific question, you mentioned Shannon Diversity Index. And for those who may not know what that means as far as diversity goes, what is the Shannon Diversity Index? Like, what does it represent?
So the Shannon Diversity Index, like it sounds, is a measure of the diversity of certain communities, and it takes into account kind of what groups are present as well as their abundance, and and it turns that into a number which measures the diversity of your system.
Okay. And then my last question is, I remember when, you're doing the statistical analysis, the significant p-value and the low r-squared was kind of a surprising result. Why do you think that was? Like, why do you think we got such a low r-squared value when we used such a large data set?
I believe that could have mainly come from the fact that there are lots of different environmental factors that impact benthic invertebrates, like dissolved oxygen levels and pH and temperature, that I didn't take into account in my experiment. So while salinity did account for some of the variance in benthic invertebrate diversity and abundance, other factors could have also contributed, and the interactions between those factors could have also made some differences as well. So I believe that was the main contributor to my low r-squared value.
Okay, great. Thanks. Good job.
Thank you.
Awesome. And we've got one other question from the audience. You can just kind of see if there's any advice that you would have for any other students who are interested in research.
Yeah, sure. My favorite part of the research experience actually was going out to sample. I originally was doing an experiment where I would be sampling invertebrates, and I'd say that was a very fun way to spend my summer. As for advice, it's kind of like what I said before. You're going to face a lot of challenges that you won't expect in the research process. So just try to be persistent and get creative, and you'll probably end up researching in your general area of interest and still ending up with a project that you're really happy with.
Awesome. And I'm glad to hear that you're very happy with your project because you should be. You did a fantastic job.
Thank you so much.
Yeah. All right. Thank you both. We have one more student, and I actually have the pleasure of introducing Adi. Unfortunately, his mentor Danny couldn't join us for this session, but I do have some notes from him. And so Danny said that Adi was great to work with. He took on a really challenging and entrepreneurial project, and his ambition allowed him to collect patient data and build a useful software tool that has the potential to make a really positive impact on people suffering with hyperkalemia. And Adi, Danny said you were a pleasure working with this summer, and he very much enjoyed getting to know you. So with that, I would love to have you tell us about your project.
Alrighty, thank you. So my name is Adi Ben-Stadia. I am a sophomore at Pineview School here in Osprey, Florida. And over the summer, I worked on developing a home-based, non-invasive approach for hyperkalemia detection. So the first thing you're probably wondering is, what is hyperkalemia? So hyperkalemia is basically an elevated level of serum potassium or too much potassium in the blood. So for the purposes of this study, we're going to define it as 5.2 milliequivalents per liter. And some symptoms include muscle fatigue, weakness, paralysis, arrhythmias, and or nausea. So it's really not a great thing to have. So next thing you're wondering is, who's at risk? The short answer is a lot of people are, because it affects about 6.3% of the general population. The long answer is that individuals with diabetes, chronic kidney disease or CKD, substance abuse history, and or heart disease are most susceptible to hyperkalemia. The problem, though, is hyperkalemia monitoring. So patients who have hyperkalemia have to undergo frequent blood work to monitor their condition. And that frequent invasive blood work is a significant burden on the patient in terms of both health and finances. It poses the risk of hematomas and significant blood vessel damage. And patients must also arrange for transportation, and frequent monitoring costs them over $8,000 more in healthcare costs compared to other patients. So what we want to study is a way to detect hyperkalemia non-invasively. So our line of reasoning is this. Potassium is an electrolyte, so any fluctuations in it, like hyperkalemia, are going to have a significant impact on the electrical system of the heart. An electrocardiogram, or an EKG, is a common way to view the electrical signals of the heart, which means that hyperkalemia should have a significant impact on the patterns of an EKG, such as the peaked T waves that you can see in this figure on the right, a wider QRS duration, and a wider PR interval. So this is not only something that we thought, it's actually something that's been proven by a lot of studies, that these characteristics show up time and time again in the EKGs of hyperkalemia patients. So we hypothesized that EKG can be used as a suitable hyperkalemia detection and serum potassium estimation method. So the knowledge gap, several studies have explored using artificial intelligence or AI to detect hyperkalemia from EKG data. A lot of those studies have extremely impressive accuracies, such as ECG12net that has an accuracy of about 97%, and others don't, like Galloway and colleagues at I think that was sometime in the early 2000s, right when this technology was in its infancies, and they had an accuracy of about 50%. But none of those studies really fit our design requirements. Most of them require 12 lead EKGs, which aren't accessible to patients at home. At home, patients will probably have 1 lead EKGs. And they're all extremely computationally intensive, meaning that patients and low resource departments are not able to use them. So we need to develop a computationally lightweight model that can be used by patients at home to detect hyperkalemia from a single lead EKG, which is easily accessible. So here's our approach. The first stage is data set collection. We decided to use an Apple Watch Series 6, which is, as many of you know, is a very widespread device that has an EKG monitor or an EKG sensor in it, and it's accessible to all patients. So we use that to collect EKGs from hemodialysis patients. The reason why we use dialysis patients is because one, they have blood work going on in real time, which means we have a temporally accurate serum potassium level to correlate the EKG to. And two, they have a higher incidence of hyperkalemia, which means that we can get more hyperkalemia training data from a similarly sized patient data set. So we took about 4 to 5 EKGs from each patient at a hemodialysis center here in Sarasota. The second stage was creation of the image recognition model. So we used a software called Teachable Machine that's developed by Google. It's readily available online, and it is a very convenient way to create image classification models. So once I input all of the training data that I collected into the image recognition model, then what I can do is quickly train the model, and then I can test different EKGs in that model, and for each EKG, it will give me a decision confidence. That decision confidence is based on the confidence that the model has that the EKG displays hyperkalemia. So if that number is 0%, that means the model is confident that that is a normal EKG. And if it's 100%, that means that that model is confident that it is a hyperkalemia EKG. And then there's a lot of gray area in between, obviously. And then stage 3 is the creation of the hyperbolic tangent regression model. So basically what happened is I used the confidence that I just discussed before and trained a hyperbolic tangent regression model to be able to output the exact serum potassium level. Because while it might be helpful for patients to know whether or not they have hyperkalemia, obviously clinicians will need to know a lot more information. They'd want to know the numerical potassium. And so that's why we're trying to estimate the numerical potassium level from the image recognition model. So first, I want to go over the patient data set characteristics. So a total of 11 patients volunteered for the study, which yielded 54 EKGs. All of the EKGs were collected at the U.S. Renal Dialysis Center here in Sarasota, Florida. The median potassium level was 4.8 milliequivalents per liter, and the mean was 4.83 milliequivalents per liter, which is classified as normal kalemia. That means that the average patient had a normal serum potassium level, but about 36.3% of patients did have hyperkalemia, and that's similar to a lot of previous studies that put that anywhere between 25 and 30%. So this is a very healthy and seems to be a robust data set for training the model. So next, we're going to go to the validation of the EKG classification model, the image recognition model. So the training data set contains 46 EKGs, and the testing data set contains 8 EKGs. Out of the testing data set, 5 of them were normal, and 3 EKGs were hyperkalemia. So on that figure on the left, we can see that the model trained in 7.56 seconds, and that figure is what we call a confusion matrix. That means that basically what this one is showing is that the model has a self-computed accuracy of 100%, so no false positives or negatives. You can see that the top right corner and bottom left corner are both 0. Then the next graph in the middle is the accuracy per epoch graph, and that's showing how the accuracy changes each time the data is cycled through the model. So 100% accuracy was reached at about the 16th cycle during the training of the model. And then the final graph is the loss per epoch graph, and that shows the loss or the difference between the predicted and actual results changing each time the data is cycled through. So loss will continue to decrease until the 50th cycle, where it is 0.0501, which is very low loss, which is good. So the results of the hyperbolic tangent regression model, as you remember, this is the serum potassium quantification approach. The confidence percentage is very low for normal EKGs, as we can see on that scatter plot on the left. So everything falling below 5.2 milliequivalents per liter has a very low hyperkalemia confidence, which is ideal. And then anything above 5.2 milliequivalents per liter, which we can also see in the scatter plot, has a very high confidence, hyperkalemia confidence, decision confidence, sorry. So that is also an ideal result. And next, I created the hyperbolic tangent regression model that you can see on the right. It's superimposed on a scatter plot that we saw on the left. And the scatter plot of the decision and serum potassium level, also you can see visually that this seems to be a very good fit. And it has an R-squared value of 0.9989, which suggests that the hyperbolic tangent regression model can explain 99.89% of the variability in the data between serum potassium and confidence. And that suggests that it could be a very accurate and very effective way to quantify the serum potassium from those decision confidence. So the discussion, what are the big takeaways from this project, both in the field and outside of it? So this is the first study to date that provides a non-invasive hyperkalemia detection that is ideal for home use. So it uses a one-lead EKG and it's computationally lightweight. It can be used at home by patients. It can be used in low-resource hospital departments. Our two-stage approach is also one of the few EKG-based hyperkalemia detection systems to estimate numerical serum potassium. That's important, obviously, for clinical applications because you want to have that numerical potassium estimate. Next, we used a novel EKG analysis approach. So currently, most studies that analyze EKGs for any condition work by extracting features from the raw EKG data for analysis. That is a highly effective approach, but it's also very computationally demanding. So this study demonstrated that just simple image recognition can also be a very effective EKG analysis technique. You know, we saw how effective it was at classifying hyperkalemia, so it might be effective for also other conditions. And a large impact on patient care. So if we develop this even more in the future, patients may be able to track their potassium at home as frequently as needed with no invasive blood work, which has a host of benefits for the patients. You know, better condition of their blood vessels for future procedures, lower risk of hematomas, things like that, and also a lower financial burden on them and the healthcare system. And finally, I want to just talk about some potential improvements. So a huge improvement is increasing the size of the data set. You've probably heard this. Any AI model is only as good as the data that you put into it. This was 54 EKG data set, which is much larger than what I've worked on previously. Previously, when I worked on hyperkalemia detection using EKGs, I had a data set of maybe 20 or 25 EKGs. So this is a significant increase from there. Obviously, it's very difficult to find and collect a data set that correlates these two. But in the future, if this can be done with even more EKGs to analyze, that would definitely increase the effectiveness of this model. So some quick acknowledgements. We'd like to thank the U.S. Renal Dialysis Unit in Sarasota for allowing me to collect data from their hemodialysis patients, as well as the patients themselves for collecting their EKG data. It's a very important aspect of this project. And finally, we'd also like to extend our gratitude to the Headwaters Research Institute. So thank you to Danny, Sean, Megan, and everybody else for all of their guidance and advice in terms of putting the project together, putting the presentation together and the manuscript. And so, yeah, thank you all so much. And I think that should be it.
Awesome. That is fantastic work. I think the first question that I have for you is, it sounds like you've worked on this a little bit before, but what got you started in this area? What made you want to research this?
Yeah, so originally the reason why is because my grandmother has hyperkalemia. And so that's kind of like my original starting point. And she had to do a lot of very frequent blood work. And so, you know, she had like hematomas and other complications because of that frequent blood work, as well as, you know, one of my aunts always had to drive her to the office to get blood collected. So that was definitely probably a big source of inspiration for me, is seeing that problem and wanting to solve it.
Nice. And so a couple other questions for you. Do you have ideas for other like are there other opportunities to use data from wearables like Apple Watches and other devices that we could do this for, so people don't have to travel to get blood work or other tests done?
Oh yeah,
Have you thought
definitely.
about that?
Yeah, so if we're talking specifically within the realm of EKG sensors, there's huge potential, because there's a lot of electrolytes in the body. Potassium is just one of them. Sodium, there's lots of electrolytes, and any imbalances of them can cause differences in the EKG. So for sure, these kind of wearable EKG machines, portable EKG machines, can be very useful in the future detection of these, and taking a similar approach with stages like image classification and then quantifying them in a regression model. That's definitely a very exciting approach, and could be very promising for lots of other conditions.
Awesome. And then final question from me is, are there any other, as you're thinking about your data and as you're thinking about potentially having bigger data sets and training your model to search for this, are there any other like afflictions that can look like hyperkalemia, or anything that might start to confuse your data, or comorbidities or something that you have to kind of control for and watch out for as you keep expanding?
Yeah, so there are certainly a few. So things like a broadened QRS complex, or a broadened PR interval, and those are definitely a lot of other conditions have those. I would say that the biggest kind of telltale sign of hyperkalemia is that peaked T wave, because odds are if there is a peaked T wave, then that will be a case of hyperkalemia. So I think that's really the biggest feature of that is unique, or somewhat unique, to hyperkalemia. But yeah, there are definitely other diseases that could be confounding.
Awesome. And then I just wanted to share this with you, that we've got some, thank you for representing Sarasota from Sarah. Just a quick note from your neck of the woods.
Thank
that
you.
you did fantastic research. And yeah, it looks like we don't have any other questions. So I just want to end with, was there anything that you found surprising, especially as you were coming through the like Headwaters research process and going from start to finish with our questions and things, even though you'd been thinking about it and done a lot of research before? Was there anything different about, this time around?
Yeah, I think there was, especially in terms of of course, more advanced data analysis techniques, things like that. Putting together the manuscript, especially because last time I was working on this, I didn't put together a manuscript. But yeah, those are definitely added to the experience. Yeah.
Awesome. Great job. Well, thank you so much, and great work, and looking forward to seeing where you take this in the future, because I think your future studies are going to be just as impressive.
Thank you. Thank you so much.
All right, great. And I just wanted to wrap up by bringing Sean up here, my partner in teaching this summer, and we wanted to say a couple words. First, thank you, Sean, for helping. Thank you to all of our mentors who were amazing this summer. We had, as you saw, such varied projects, and the mentors, some of them have expertise in those specific areas. Some of them are learning right alongside our students. And so that's always a really fun challenge, and everyone really stepped up to the plate. But the students were all fantastic. And yeah, thank you, Sean, for joining us for this. Do you have anything to add?
No, I mean, I think I speak on behalf of all of our mentors when I say, we all enjoyed working with each of our students. Obviously, Megan, you and I got the joy of working with each student, and when we're lucky enough to work with amazing kids, like, we get to see just a bright future for them. And I think that's the biggest joy for us as mentors, and just working with, young minds that get excited about science. So I think, like I said, we see bright futures ahead for all of them. So it's been a pleasure. So
Yeah,
honestly, I thank you, students, right? I mean, you guys made it easy.
Yeah, and you came up with such intriguing questions and things that, you know, are totally outside of what I would have been thinking about for a research question. And yeah, it's been really fun. And we're excited to do it again in the fall. So if anyone knows any students who might be interested, we do have some space in the fall research experience. You can find that on the website as well. But we are also looking forward to seeing these finished manuscripts from all the students. They'll be published in the fall, and looking forward to seeing where everyone goes with all of their science work. So thank you all for joining us so much. And the recordings will be up online shortly right after this.